cytomegalovirus. These are other common causes of pharyngitis. Croup, this one is going to talk about it in a second again, also called acute laryngeotracheal bronchitis. Okay, this is a specific viral infection of children under 3 years old. Very important characteristic feature of croup is what's described as a barking cough, or seal-like cough, and acute inspiratory stridor. Okay. The other one that's important here is bronchiolitis. This is also a distinct syndrome of infants and young children. Okay. And then we have viral pneumonia. Viral pneumonia is basically described as dysfunctional gas exchange accompanied by inflammation of the lung parenchyma. Okay. Dry cough, rhinitis, and conjunctivitis are the most common presentations. How do viruses reach the small airway? They reach it through continuous spread or by inhalation of aerosols or respiratory droplets, which is what happens with the coronavirus. Okay. After that, the mucosal infection will destroy the epithelial cells. Okay. Viral causes of pneumonia includes measles, varicella, hantavirus as well. Okay, so let me talk about the lower respiratory system now. We're done with the upper. So influenza. The signs and symptoms of the flu or influenza include a sudden fever accompanied by pharyngitis, congestion, a dry cough, malaise, headache, and myalgia. Okay. Most people, they recover within 2 weeks. Okay. The influenza virus, you guys have to know about the influenza virus. It's an enveloped, segmented, negative sense, single-stranded RNA virus that has a helical capsid. Okay. The influenza virus has 2 species designated as type A influenza virus and type B influenza virus. Okay. The mode of transmission of the flu virus is through respiratory droplets. Okay. Through respiratory droplets. Now, the influenza virus is has a segmented genome with about 8 RNA molecules surrounded by a lipid envelope that has glycoprotein spikes. 2 important ones are named hemagglutinin and neuraminidase. These spikes help the flu virus to attach to our lung cells. Okay, so imagine it like this. It has an envelope and it has 2 spikes, hemagglutinin A and NA, neuraminidase. Okay. Now, this next part is important. Mutations in the genes that code for these glycoprotein spikes is what's responsible for producing new strains of the influenza virus. Remember, whenever you get the flu shot or the vaccine, it's only defending you against the most common influenza strains, not all of them. Not all of them, okay? So there's 2 important concepts here. One is called the antigenic drift, okay? This refers to accumulation of hemagglutinin and neuraminidase gene mutations in a specific geographic area, Whereas antigenic shift is this is a major antigenic change. This results from reassortment, genetic reassortment of different types of influenza viruses infecting the same cell. We're going to talk about this in a second. So, anyways, any type of strain of the influenza virus is identified by the HA and NA antigens. Okay? Mode of transmission, primarily through inhalation of respiratory droplets, but also can be transmitted by contaminated fomites as well. Okay? Pathogenesis. The influenza virus has tissue affinity or tissue tropism to our respiratory epithelial cells. Okay? Respiratory epithelial cells. As we said, we have two distinct glycoprotein spikes, HA and NA, and then anchoring the bases of each of these spikes on the inside of the envelope is a M protein called membrane protein, and you guys can see it in this picture. Okay, this is the membrane protein right here. See how it anchors the two spikes, the two HA and NA spikes. This is the segmented RNA. This is the lipid bilayer. Okay. Now, HA, this attaches to our cells, specifically attaches to sialic acid receptors. Sialic acid receptors are on our cells, okay? Sialic acid receptors are present on the surface of erythrocytes. So viruses with HA glycoproteins are therefore able to cause heme agglutination when mixed with red blood cells. Sialic acid receptors are also present on the upper respiratory tract cells. And the hemagglutinin receptor binds to these cells, okay, activating fusion of our plasma membrane with the influenza virus plasma membrane, which then results in the release. You guys remember the release stage of the viral infections? It's going to release it into the cell of the our cells. So that means that hemagglutinin A is basically needed for the first step of the viral cycle, which is the adsorption or attachment. Okay. So whenever you have antibodies, for example, from a vaccine, the antibodies, what they do is that they're blocking this binding and thereby preventing infection. Okay. Now, what is neuroaminic acid? Neuroaminic acid, okay? Neuroaminic acid, this is a important component of mucin. Mucin or mucin is a basically a substance that covers our mucosal epithelial cells, okay? And it makes up an important part of the upper respiratory tract defense barrier. It's part of the innate immune system as a defense barrier, okay? So neuroaminidase, this is an enzyme. What it does is it cuts it or cleaves the neuroaminic acid and thereby disrupts the mucin barrier. When it disrupts it, it will expose the sialic acid receptors that are lying underneath the mucus barrier. So you disrupt the barrier. When you disrupt the barrier, you're going to expose the receptors. Once those receptors are exposed, the influenza virus is able to attach to it and cause an infection. So remember, know these two roles, okay? So influenza, any questions so far? Are you guys following me?